Morning, all of you.
Start,
All
sir.
of
Start.
okay. All of you, good morning. So yesterday we completed the glycolysis and oxidative decarboxylation of pyruvic acid also. So today just uh, yesterday completed the chapter. What is that oxidative decarboxylation of pyruvic acid? We will discuss again. So in this, so okay, oxidative pyruvate. So in this, so three carbons of pyruvate undergoes to the what is that oxidation reaction. In these, NAD plus is converted into NADH plus. These are called as dehydration or what is the dehydrogenation here, NAD plus and converted into NADH. And in this, one carbon dioxide is comes out. So this process is called as what is that decarboxylation. So dehydrogenation and decarboxylation. One carbon molecule is comes out. So that's why remaining how many are there? Here pyruvate is three carbons. So that's why one carbon dioxide is removed. So here next to form the what is that acetyl coenzyme A. So acetyl coenzyme is contain the what is the two carbons. So the three carbon of pyruvate is oxidative decarboxylation undergoes to the oxidative and decarboxylation and then formed the two carbons of acetyl coenzyme A. So here equation the equation purpose you observe here the pyruvic acid. So coenzyme A here coenzyme A is added to here condenses with the coenzyme A. So what is that? The pyruvate acid is condenses with the coenzyme A and here formed the what is that and dehydrogenation. NAD plus is converted into NADH and one carbon dioxide is removable. So that's time here formed the what is that acetyl coenzyme. Here you are observing the pyruvic acid and condenses with the coenzyme A and the NAD plus and converted into what is that acetyl coenzyme A and CO2 is removal. So here formed the what is that NADH plus H plus. So this reaction is catalyzed by the what is that pyruvic dehydrogenase enzyme. So understand better here the three carbons of pyruvic acid is converted into the two carbons of acetyl coenzyme A. So in the presence of what is that pyruvic dehydrogenase. So this reaction is called as what is that? This is the what is that? This is the represent the second stage of the aerobic respiration and occur in the mitochondrial cortex. So here in this two molecules of pyruvic acid is generally end product is what is that in a glycolysis two molecules of pyruvic acid. So here also two molecules of pyruvic acid is converted into the two molecules of acetyl coenzyme A. So acetyl coenzyme A is the here which is very popularly known as the connecting link between the glycolysis and the Krebs cycle. So that is the important you understand better here. Acetyl coenzyme is the what is the coenzyme A, which is very popularly known as the what is that connecting link between the glycolysis and Krebs cycle. Okay, now energy sheet of oxidative decarboxylation of pyruvic acid. So here the pyruvic acid to converted into what is that acetyl coenzyme A. In this, so already we know what is that here NAD. So two molecules here taken the only one, but you remember it what is the two molecules. So that's why here also two molecules. So one NAD is already you know what is that one NAD is what is that gives rise to the one ATP, three ATP. So here two molecules are there. So two NAD converted into two NADH. That's why here formed the what is that? How many ATPs? Two. Two means six. One NADH is equal to one NADH here two NADH are there, each worth is 3 ATP. So that's why in the energy sheet of oxidative decarboxylation of pyruvic acid, net gain ATPs are 6 ATPs. That is, remember it. So 2 NADH are undergoes to dehydrogenation reaction. So that's why formed the 6 ATP. Okay, then now Krebs cycle here. So in a 1937, English biochemist Sir Hans Krebs postulated the cycle of reactions which described the oxidation of pyruvate to carbon dioxide and came to be known as a citric acid cycle. So in 1953, Krebs was awarded Nobel Prize and the cycle is frequently referred to as Krebs cycle in his honor. So it is also known as TCA cycle. So TCA cycle means tricarboxylic acid cycle. So it consists of three COOH groups. So that's why it is called as TCA cycles. And another one name is what is that citric acid cycle. Citric acid cycle means the first formed substances in this cycle is citric acid. Hence, it is also known as the citric acid cycle. So let us now observe this cycle. So here beta Krebs cycle is what is that first here acetyl co-MgMA. So acetyl co-MgMA is formed from the which one glycolysis. Already we know. So glycolysis end product is what is that pyruvic acid. So this pyruvic acid is undergoes to the oxidative decarboxylation process and end product is released the what is that acetyl co-MgMA. So which contain the two carbons. So this is the acetyl co-MgMA. So these two carbons acetyl co-MgMA condensed with the with the condenses with the what is that oxalo uh, oxaloacetic acid or so then after here what is that formed the what is that citric acid citrate first one. So these are the four carbons. So it is the four carbons and is the acetyl co-MgMA is two carbons. Total how many carbons are there? Six carbons. So these six carbons citrate is formed. So that's why first stable compound is first formed substance is citric acid. So citrate. So that's why this cycle is called as citric acid cycle. So these are the process is called as condensation. And here then reaction is the dehydration. So in a dehydration, what is that citrate? So our citric acid is so converted into the cis aconitate. So cis aconitate or cis aconitic acid. So here water molecule is removed from these in this reaction. So this reaction is catalyzed by the aconitase enzyme. So this is the water is removed from the citric acid. So this reaction is called as dehydration. Next one hydration. So this reaction is next reaction is hydration. In this reaction, cis aconitic acid or cis aconitate. So water is comes so enter into these substances. So these are called as what is that hydration. So this is reaction is above reaction is called as what is that dehydration. So this reaction is called as hydration. So it is catalyzed by the aconitase, similar enzyme here also. Dehydration and the hydration, both reaction involve are catalyzed by the which enzyme? One aconitase enzyme. So it is also six carbon substances, cis aconitase. Next, this cis aconitic acid and converted into isocitrate. So these process water is combined or joined in this cis aconitic acid to and isocitrate. So this reaction is called as what is that hydration. So here beta, so you are observing isocitric acid or isocitrated there in these. So oxidation one reaction, you are observing NAD plus. So here in this reaction NAD plus is there reduced to what is that NADH plus H plus. So NAD plus reduced to what is that NADH plus H plus. And also here CO2 also comes out, will be come out. So here CO2 is removed outside. So that's why it is called as what is that decarboxylation. Carbon dioxide is removed and here what is that NAD plus is reduced to what is that NADH plus H plus. So that's why these first reaction of the oxidation. So you notice that it is the what is that first oxidation and first one decarboxylation. It is remember it better. So these are isocitrate is converted into alpha ketoglutarate or alpha ketoglutaric acid. So it is catalyzed by the isocitrate dehydrogenase. So which enzyme are catalyzed? Isocitrate dehydrogenase. So here beta you are observing oxalosuccinic or OAA. So here what is that? This is the acetyl coenzyme here, 2 carbons and 4 carbons gives to the citric acid 6 carbons. So remaining all substances are contain the what is that? So 6 carbons. But here these are isocitrate also 6 carbons. But here carbon dioxide is removed. One more carbon dioxide is removed. So that's why one carbon is removed. So here 6C2 the converted which is formed the alpha glutaric acid, it contains the 5 carbons. So why? Because here one carbon dioxide, one carbon is removed, so remaining how many carbons are there? 5 carbons here there. So that's why the alpha glutaric acid contain the 5 carbons. So here you are observing beta. Next one, al here alpha glutaric acid to succinyl coenzyme A. So here also you are observing. So NAD plus is what is that? In this reaction also NAD plus is reduced to NADH plus H plus, and here also 1 mole of carbon dioxide is removed. So that's why already alpha glutaric acid is what is that? 5 carbons. So here one carbon dioxide is removed. So that's why remaining how many carbons are there? 4 carbons. So these 4 carbons are succinyl coenzyme is formed. So here also second one what is that? It is the oxidation one, and here also decarboxylation. So then reaction succinyl coenzyme A, and you are observing. So here the what is that? The ATP is formed. So it is like a what is that? Substrate level phosphorylation. So in the Krebs cycle in animals, mitochondria produce the GTP instead of ATP. So that is the important. So what is that? The Krebs cycle in animals, mitochondria produce the GTP. Here GTP. So instead of ATP. So here coenzyme is the succinyl coenzyme is cleavage as what is that? Coenzyme A outside, and the form of the what is that? Succinic amla. So acid succinate. So then here you are observing the succinate is converted into, and these are involved into the oxidation reaction. So but he in this oxidation there here FAD is reduced. Here what is that? FAD is reduced to FADH2 or FADH plus H plus. So and convert these succinic acid is converted into fumarate. So in this reaction, the specific catalyzed enzyme is succinic dehydrogenase. So here first these cleavage reaction is, so succinyl coenzyme to form the succinic acid and coenzyme A. These in these reactions catalyzed by the enzyme is succinyl coenzyme synthetase or thiokinase also involved. So here succinic dehydrogenase in the presence of the succinic acid is converted into the fumaric acid or fumarate, and then the fumarate is converted into the water, and again join with the substratum what is that? Fumarate in the presence of enzyme fumarase and form the malate or malic acid. So here malate and the here the what is that undergoes to the again oxidation process in these NAD plus and the NADH plus H plus. So these are catalyzed by the malate dehydrogenation. So here oxalosuccinic acid or alpha OAA oxaloacetic acid also formed. So both are the what is that? 4 carbons. So that's why these cycles is continued this in the 2 times. So why? Because in a glycolysis uh, two end product is formed the what is the 2 molecules of pyruvic acid. That's why here formed the 2 molecules of acetyl coenzyme A. So this cycle is completely rotated or continued process in the two times. So that's why all are the two molecules converted. Here 2CO2, here 2CO2, 2 NAD. So here this is. So as a reactions, we can first understood the condensation. In a condensation, what is that? So oxaloacetic acid, so oxalosuccinic acid also similar. Oxaloacetic acid plus and the acetyl coenzyme A in the catalyzed enzyme is what is that? Citric synthetase, and formed the what is that? Citric acid plus coenzyme A. So here four carbons and it is the two carbons. So first formed the acid is six carbons. So that's why we are giving the name as also what is the citric acid cycle. So then after second one condensation process. Next one what is the dehydration. So in a dehydration, what is that? The citric acid. So catalyze citric acid is converted to what is the cis aconitic acid and the water molecule is removal. So this is the aconitase enzyme. It is catalyzed by the aconitase enzyme. And next one hydration. So in hydration, next process, what is that? Cis aconitic acid catalyzed in this reaction catalyzed by the enzyme is aconitase, and cis aconitic acid is converted into isocitric acid. So clearly you observe better here what is that? Cis aconitic acid and isocitric acid. So that is the important. And next one oxidation. So isocitric acid is the involved into the oxidation process, means the reaction NAD plus is reduced as what is the 2 as a NADH. So isocitric acid and converted into the oxalosuccinic acid in the presence of isocitric dehydrogenase. And next one, so decarboxylation. So in a decarboxylation, the sixth carbon of oxalosuccinic acid release one molecule of carbon dioxide in the presence of the oxalosuccinic decarboxylase. So it formed the what is that? Alpha ketoglutarate acid and remove the carbon dioxide. So next one, oxidative decarboxylation. This is also known as oxidation 2. So in this alpha ketoglutarate acid plus NAD plus coenzyme A is in the presence of alpha ketoglutarate dehydrogenase formed the succinyl coenzyme A, NADH plus CO2. So this is the sixth reaction, oxidative decarboxylation or oxidation 2. And next one, cleavage. So in this cleavage, so succinyl coenzyme A, so is what is that? ADP and inorganic phosphate, means here ATP is synthesized or ATP formed. So these are also known as this. In this we can get the what is the succinic acid and succinyl thiokinase and formed. Next one, oxidation 3. So in this succinic acid is what is FAD. In this FAD is reduced to the what is that? FADH2. So in the presence of succinic dehydrogenase, fumaric acid formed the fumaric acid plus FADH2. And next one, hydration. So hydration means, so water is added. So fumaric acid combined with the water molecule in the catalyze. This reaction is catalyzed by the fumarase and formed the what is that? Malic acid. So next one, what is that? Oxidation 4. So next malic acid is what is that? In this reaction, same similar. NAD plus is reduced to NADH plus H plus, as here a malic acid here NAD plus in the presence of malic dehydrogenase and form the oxaloacetic acid. So these are the 10 reactions of Krebs cycle. So here energy sheet you understand first carefully. So just wait, you are observing here. So oxidations process you are here, you are clearly observe better here. So one NAD plus NAD plus sorry, NAD plus give one reaction, second reaction. So here you are observing three reactions. So how many times this cycle can rotate in the two times? So what is that? Two times one, two, four, and five, six. So here also, so two FADH2. So total NAD, how many are oxidations? One, two, and here 3, and FAD is 1. It is also oxidation process. So here this is the what is that? Substrate level phosphorylation. So remember it. So how many are there? 1, 2 and 3. So FAD also oxidation. So total 4. So but here NAD. So NAD. So here energy sheet observed. So ATP produced the what is that substrate level phosphorylation. So where the succinyl coenzyme to succinic acid. So how many ATPs are formed? Two ATPs. Next, ATP formed from the NADH. So the reactions is what is that isocitric acid to oxalosuccinic acid. Here also two reactions. So each NADH is the what is that one NADH is equal to 3 ATP. So here two types rotating two types are there. So that's why 2 into 3. So 6 ATPs. And next one, what is that? Alpha ketoglutaric acid to succinyl coenzyme A. Here also 2 into 3. So total how many? 6 ATPs. And next one, what is that? Malic acid. Malic acid to oxaloacetic acid. Already I'm now we have to know. So NADH oxidation process, how many places are there? Three places. So that's why you are observing three places means each one places two. So here, so each one places three ATPs, two times. So 2 into 3, 2 into 3, 2 into 3. So here 6 ATPs, three place. Next one, what is that? FADH2. So FADH2, what is that? Where these reaction? So succinic acid to fumaric acid. So that places is already we know one FAD is equal to 2 ATP. So that's why two times. So 2 into 2. So total how many ATPs are formed? 4 ATPs are formed. So here total. So total what is that? 6 12 6 18 plus 20 plus 24. In a Krebs cycle, net ATP gain is what is the 24 ATPs are there. So simple. So it is not necessary if you have to the full knowledge on the diagram or flow chart. Immediately you can calculate it here also fastly. So you are observing two times. So 2 and the 2. How many are there? 2 to 6. 6 into 2, 12. Okay. Here 3 3 6. Here 6. 6 6 12 plus here also 6 8. Okay. Here also 4 20. So okay. So this is the process for the easy remembering this. NADH is how many places? 1 2 3, and next FAD, and this is so. This is the balance sheet of the uh, Krebs cycle. So let us now we discuss the electron transport system. So in a electron transport system, so electron transport system. So first one, this electron transport system is, so this side is the matrix place, and this is the outer side is the what is the inner membrane spaces. So these electron process, so this is the dependent process occur in the inner mitochondrial membrane. This process involves the five complexes. Here you are observing 1 and 2 and 3 and the 4 and the 5. So in these total how many complexes are involved? Five multi protein complexes are involved. So first one, second one, and third one, and fourth one, and fifth one. So in these first one, component or complex one. So this is the complex one. It is the NADH ubiquinone oxidoreductase actually. So oxidoreductase. It is a complex enzyme having a flavin mononucleotide. It contains the FMN. So flavo mononucleotide FMN as a prosthetic group. So these contain the enzyme is also contain the iron and sulfur FES protein. And next one, complex 2. So here complex 2 means succinic acid. So succinate here. So in a Krebs cycle, FAD is oxidation. We can observe the what is that succinyl coenzyme A to succinic acid to fumaric acid formation. The FAD is oxidized. So that's why here succinate. So here these in this group in this complex 2, prosthetic group is FAD. in this prosthetic. In this prosthetic group is FMN. In this is FAD. And here Q is there, like in a photo electron transport like plastoquinone like structure. So it is called as ubiquinone. It is a lipid soluble quinone which can diffuse freely in the mitochondrial membrane. It is a structure similar to the plastoquinone found in chloroplast. It is the mobile carrier. It is the mobile carrier. So between the complex 1 and 3, between the complex 1 and 3. So, and the next one sometimes also 1 and third, and also between second and third. So, so this is the important. So sometimes here, sometimes here also present. So next complex third. So complex third is what is that cytochrome C. So these are called as cytochrome reductase. So these contain the complexes cytochrome C, and it contain the what is that one iron and sulfur protein. So next one complex 4. So complex 4 is cytochrome C complete. Cytochrome C oxidized there. So last one is the complex 5. So these complex 5 is called as what is that ATP synthetase. So here you are observing electron transport. So this process involve what is that 5 multi-protein complex along with the mobile carrier. Mobile carrier is what is that ubiquinone and cytochrome C. So in this complex 1, so in a complex 1 prosthetic group is what is that FMN, and complex 2 prosthetic group is what is that FAD, and another one ubiquinone is the what is that ubiquinone mobile carrier. It is the mobile carrier between complex 1 and complex 3, and also between sometimes complex 2 and the complex 3. So next one, complex 3. So in this complex has the cytochrome B and cytochrome C1. It contains the iron and sulfur. And next one, cytochrome C. It is a mobile carrier between complex 3 and complex 4. And next one, complex 4. So here these enzyme contain the cytochrome A and cytochrome A3, and these are contain the 2 copper containing proteins. This complex transfers electrons from reduced to cytochrome C to molecular oxygen. So here complex 5. So complex 5 is the what is that ATP synthesis. It contains the 2 parts, that is the F1 and F2. So here these complexes contain the F1 part is also known as headpiece, and F0 is the base piece. So this is the structure of the complex 5. So now let us we discuss about the process electron transfer. So first in a complex 1, this is the what is that matrix, and it is the inner membrane spaces. So complex 1. So this complex 1 is considered as the gateway for electron transport. What is that? Gateway electron transport. Here, uh, 2 electrons from each NADH and the generated into mitochondrial matrix places. These are the mitochondrial matrix places from oxidate to decarboxylation of the pyruvic acid, and this is the Krebs cycle. Okay. Next are transferred to the, here what is the prosthetic group is FMA. So next the electrons complex substrate. These electrons are two electrons are transformed, and various opinions are there pumping the protons to outside inner membrane spaces. So there is the mainly different opinions are there. So but here the every 2 electrons passing through complex 1, there is in the pumping of 4 protons. So 4 protons from the matrix into the inner membrane spaces. So that is the important. So then these electrons is comes to here are ubiquinol, and then electrons are ubiquinone is the what is that a proton pool. So when these electrons are transferred to the cytochrome C, so B complexes, then immediately remaining these electrons loss is filled with the what is that FADH to FAD. So these are from the complex 2 is fulfill the loss of the electron transport to the complex 3. So that's time each every 2 electron transport, so pumped out the 4 protons to the outside of the inner membrane spaces. Here also complex 4 places also uh, complex 4 also released the what is that, how many protons are outside. So 4 protons, and then these means here what is that, cytochrome b, cytochrome c here. Then electrons is transferred to the cytochrome c. So it is the linker between the sorry, complex 3 to complex 4. Cytochrome c is there. Then electrons is comes to complex 4. It is the complex a1 and a3 here, a1 and a3. Then here only how many, already we know what is that, total 2 electrons are transferred, every 2 electrons carries and pumped out to the 4 protons to the outside the inner membrane spaces. So but here in a complex 4 places only release the 2 protons. So remaining 2 protons are added to the what is that, oxygen. So and formed the what is that, water molecule. Means here remaining 2 protons are consumed for the formation of water molecule. So this is the end. So from the complex 1 to complex from the complex 1, so in a complex 4 end product is we can get the what is that, water molecule. So here again you understand better. First one you are observing. So first these NADH, NAD plus and FADH to FAD. So here succinic acid to fumaric acid formation. This is the oxidation of the FAD, and remaining NADH number of places are 3 places are there. So in these in this oxidation 2 electrons are transferred. So complex 1. So simply you understand what is that, remember it better. First one complex 1 you remember in mindset, FMN, sorry, complex 1 is FMN, complex 2 is FAD, ubiquinol, and complex 3 means B and C, and complex 4 is what is that, A and A3. Next complex 5 is ATP. So now we discuss what is that, you remember it. First one is the FMN and next one is the FAD, so ubiquinol. So in this what is that, B and the C, and between the complex 3 complex 4 is C, and complex 4 is what is that, A and A3. So process just simple, you should you remember it. So NADH to NAD plus formation, 2 electrons are gives to the FMN prosthetic group, then pumped out the 4 protons at the which one, complex 1 places. Then electron goes to the what is that, ubiquinol. So then electron comes to and goes to the what is that, complex 3 cytochrome B. Then immediately these loss of electron transport to the complex 3. So here deficiency is formed. That's time this deficiency is fulfilled with the complex 2 here. So immediately fulfilled with this. Here also the complex B, cytochrome B and cytochrome C, the electron transports in by the process of oxidation and reduction process. In 2 electrons here complex 3 places, the 4 protons are pumped out to inner membrane spaces. Then these from the electrons cytochrome C and the comes to the complex 4. In complex 4 is made up with the what is that, cytochrome A and A1. So these are also electrons is comes to in the process of the oxidation and reduction. So but in these places complex 4 places 4 protons are not pumped out. Why? Because only 2 protons are pumping out to the inner membrane spaces. Remaining 2 protons are combined with the oxygen molecule. So these are the involved to the formation of water molecule. So from the one NADH oxidation you are observing, one NADH oxidation, the electron transport you are observing, how many protons are released here, 4, here 4 and 2. So total how many are there, 10 protons are released. So up to 4th complex. For example, you take the FAD. So here you are FAD, total how many protons are released here, complex 3rd places 4 and complex 4 places 2, total how many, 6 protons are released. So already we know. So what is that, the every 3 protons can gives to the what is that, 1 ATP. So that's why here one NADH, when involved into the NADH oxidation and released the total protons is what is that? 4, 4, 10. So 3 protons is 1 ATP. So that's why 4, 4, total 10. So that's why formed the 3 ATP. So that's why we write the what is that? 1 NADH is equal to 3 ATP. The reason is this is. So in the during the NADH oxidation in electron transport system, inner membrane spaces is have to the what is that? 10 protons. So that's why these are involved into the formation of 3 ATP. So that's why we write the what is that? 1 NADH is equal to 3 ATP. And similarly FAD. So you are observed. FAD means what is that here? In a electron transport from the complex 2 to 4, so release the or pumping out the protons only 6. So in a complex 3 places 4 protons, and complex second places released the 2 protons. Total how many? 4 protons plus 2 protons, so 6 protons. So that's why here formed the what is the 2 ATPs are formed. So that is the very important. So 4 protons and 2 protons. So that's why we write the what is that? One FAD is equal to 2 ATP. So let us now comes to the fifth complex. What is that? ATP synthetase. So ATP synthetase here formed the what is that? ADP to, and combine with the inorganic phosphate. So here formed the what is that? ATP. So the inorganic phosphate is combined with the ADP and form the ATP. Process is called as what is the phosphorylation. So in a matrix side here oxygen is there. So that's why it is called as oxidative phosphorylation. So simple, you are observing the complex one FMN, and complex 2 is the FAD and ubiquinone, and complex 3 is what is that? Cytochrome B and cytochrome C. So this is the very important. Okay. Here, simple understood better. So simple FMN, you remember it. FMN, FAD, ubiquinol, cytochrome B and cytochrome C. Here cytochrome A1 and A3. So it is the complex ATP synthetase. Okay, better. Now we are calculated what is that NADH process. So energy sheet is so here here complex 5 is already you know. So now oxidative phosphorylation. So in this electron transport cycle you are observing here. So what is that? First one, in oxidative phosphorylation, so this is the what is that? Outer membrane side, inner mitochondrial membrane spaces. So it is the what is that? Matrix side places. So here the proton gradient is high in the inner mitochondrial membrane spaces. So it is not a permeable to the transfer the protons, so equalization purpose from the matrix side. That's time according to the Peter Mitchell chemi-osmotic hypothesis. This purpose, so the proton gradient is where is high? Inner mitochondrial membrane places. So in a matrix places, it is consists the F1 particles. They are called as ATP synthetase. So it consists the two part, that is called as it is the F1 part and F0 part. So here F0 part is the stock part, F1 part is called as head part. So when the protons are pumped from the which one? Inner mitochondrial membrane to so matrix side. So already we know what is that? Every 3 protons formed the 1 ATP. So during these process, 3 protons are transferred through the F0 part. So it is called as F0 part is called as what is that? Proton channel. So that's time these protons are transferred through the head part. The head part means what is that? F1 part. During these, so when the protons are transferred through the F0 part to the head part, F1 part, immediately that energy can, so the proton releasing energy utilized for the adding the inorganic phosphate to the ADP. So that's why here formed the what is that? the ADP, adenosine diphosphate, is combined with the inorganic phosphate. So that's why here formed the ATP. So it occur in the presence of the oxygen. So that's why it is called as oxidative phosphorylation. So this is the reason. So what is that? Here, for example, 3 protons is here formed the 1 ATP. For example, 6 protons are transformed here formed the 2 ATP. So for example, 10 protons are released the what is that total complete the formed the three ATPs. So that's why one NADH is equal to. So here write the what is that 3 protons is gives to the what is that 1 ATP, and here write the what is that ADP, adenosine diphosphate, combined with the inorganic phosphate in the presence of oxygen. So these are called it is formed the what is the ATP. This process is called as oxidative phosphorylation. So here one NADH when one NADH involved into the oxidation, the inner mitochondrial membrane places release the or pumped out the 10 protons. So that's why formed the 3 ATP. So similarly, one FADH2 oxidation process released the 6 protons and formed the 2 ATP. So here total balance sheet of aerobic respiration. So already we know beta in the glycolysis we can get the net gain ATP 6 ATPs. So in second process, second stage is what is that oxidative decarboxylation of pyruvic acid. In this also we can get the 6 ATP net gain ATP is 6 ATPs. And next in a Krebs cycle, so we can get the what is the 24 ATPs in Krebs cycle. Total in aerobic respiration get the net gain ATP is 36 ATPs. So already we know what is that one ATP is equal to 7.6 kilocalorie energy, and so calculated into 36 into 7.6 is equal to 20
Yeah, Kumar sir,
273.6
conclude, sir.
grams. Okay, sir. So this is the complete. We can get the what is that 686 kilocalorie energy in a aerobic respiration. We can release it the energy is 686 kilocalorie energy. Now we can here get the what is the 273.6 kilocalorie. So minus getting the 412.4 kilocalorie. So these kilocalorie energy
Kumar sir,
would
time
be
up.
okay would be released